Hello, good afternoon, everyone. I hope my audio is clear. Can you confirm someone?
So,
Okay. So we'll start today's lecture with a small video. I think you already have seen this before, or if not now, you may have seen it. So this is about the power of genetic engineering, where you see that the front mouse, the mouse that is shown here, that is the engineered one, the one that is behind falling back is the normal one or the wild type. So they run about the capacities of it, and this is a treadmill, and the mouse, the genetically engineered one, is still running, the one that is for Pepsi K, phosphoenol pyruvate carboxykinase, something carbokinase, something like that. I don't remember. But the point is, the engineered one has higher capacity for um, anaerobic respiration. Okay, sorry, aerobic respiration, where it has more, so that it can engage more in Krebs cycle and generate more um, energy. And you can see that it has run for almost 5 kilometers so far, and still it is not yet done, and it is running. Well, the wild type outed a long time, much earlier. So, and you also must be aware of the things that are happening in the Nobel Prize, for example. It is given for CRISPR, and we have it in the syllabus. And that is one of the reasons why I would also want to talk about transgenic animals first. So you know how things are done in general, and then later we should go into CRISPR, where we will study a two papers or so. But the most importantly, the point when we learn CRISPR, it will be about the biology and then about applications. So in the last class, I was describing about the two of the traditional methods of how genetic engineering is done, or not traditional. They are still used and very much useful. There are two methods. One is direct microinjection method, and the other one is engineered stem cell method. So a direct method is much more simple. We just directly give the gene construct into the male pronucleus and implant into a foster mother, and then analyze the pups. In engineered embryonic stem cell method, we isolate the embryonic stem cells, then we introduce the gene, and then we select for the recombinant uh, cells, and then put it into a blastocyst to generate a chimera, and then we implant into the mouse and then get a chimeric mice, and from there we should generate a complete transgenic animals. By definition, transgenic animals is that cells that an organism should have a transgene, a gene from the outside in its genome, and all of its cells should have it, right? And it should be inherited. So in genetic manipulations, there are several ways: knock-in or transgenesis, knockout, and also we have knockdown. RNAi mediated methods of or other methods of shutting down the expression of a target gene will come under knockdown. And there is one more thing we need to understand is two other things: constitutive and conditional. Conditional can be two types. One is spatially conditioned, I mean expression in a spatial matter, and the expression in temporal ways, right? Where it should be expressed is about spatial. When it should be expressed is about temporal. Constitutive is expression all the time. So I think I asked you also to have a look at homologous recombination and site-specific recombination. In today's class, we will go a little bit of this, and then we will go to positive selection and negative selection, and probably somewhere around there we will stop today. So if one can do, as I was telling in the previous slide, gene insertion can be done, that is knock-in, or gene deletion can be done. And one should be careful also about the orientation of how you want to put and where you want to put, between which genes, whether is it targeted or are you trying it randomly. Those are the two things. If I want to put this gene between these two, I should have certain mechanisms of doing it. Targeted, targeted insertions, deletions, and so on. If we are doing targeted, we should have a targeting vector where the idea is simple, where you would have already the genes like this. Say gene 2 and part of gene 3 are here, because of which this gene 2 on the genome and gene 2 on this part, and 3 on the vector and 3 on the genome are identical or homologous, because of which homologous recombination can happen, and thereby integration may happen. Similarly, one can also do replace genes with something else. Say I want to replace with 2 and 3, and this is again a targeting vector because you have homologous regions like this between 1 and 4, like in here. And then you have a gene that you want to replace 2 and 3 with, and therefore once it is done, you will have 1 and the transgene and then the 4. 2 and 3 are no longer there. One of it is about how the integration. The second thing is, first of all, how would you put the vector or the construct into the cell? The gene of interest can be put into the cell by several mechanisms, which we may discuss at a later time, and that might be involved with. I mean, it depends on. Sorry. Yeah, so it is one can do by gene fusion, cell fusion, where you take a cell that has the gene of interest and you take the target cell and fusion may occur, and then hybridoma basically, and then the gene of interest might be incorporated or retained in the hybrid cells, and that is one way of putting it. But it's a very crude method, and it's not about how it is not regulated. We are not clear about what is retained and what is not retained. The other mechanisms would be transfection using viral particles, or chemical methods are also there. Electroporation is using voltage so that the DNA from the outside is taken up by the cells. In much better ones would be like using viral mediated injection. Some of them are also there where DNA is directly injected into the cells. Okay, there are a variety of techniques, and I think there is also called as biolistic. I hope you remember that or you know it. That is, you coat a small particle with the DNA of interest and shoot it with a gun, which is using high pressure air or neutral air, something like nitrogen, for example, and then you do it to the target tissue. So some of the cells may take up these particles, and the DNA is delivered into the cell and ultimately into nucleus, and integration may happen. So basically, there are several techniques involved in integration of the genome. Integration of gene into the genome. And when we had a class on vectors, I didn't describe something on. I waited with the retroviral vectors. This makes it much more a appropriate place to discuss it. What are retroviruses? You know about retroviruses, where the retro is about their RNA, viruses, and they be constructed into DNA. And very usually they get this DNA will get integrated into the genome, right? So by default, these RNA viruses have a propensity to integrate into the host genome. So why not use them as vectors? And I hope you will be able to recollect what is neomycin resistance. It is a resistance gene against neomycin. CMV, cytomegalovirus promoter, which is a strong constitutive promoter. One can put in the gene of interest in the multiple cloning site, this one. And there are two things important for integration of this DNA, the double-stranded DNA into the genome, right? And that integration, if it has to happen, the protein, the recombinase should recognize some genetic elements. Those will be the 3' and 5' long terminal repeats. So you can think of from here till here, all this part is the viral genome. And this part is for manipulations in E. coli. That's why you have an origin of replication. And so one, you have made the manipulation as almost treating it like a plasmid. But now one has to make it as a virus. You have to make it as a viral particle, mostly because it should be taken up by the cells, and then the construct should become double stranded. You have to make, you are doing it as in the part of DNA, and when you make the viral particles, it should have been RNA, and then you get the viruses, and those viruses are allowed to infect, which within the cell will become double stranded DNA and then integrate into the genome, right? This is the whole process. For that to occur, we have BLA, that is beta lactamase, what is for selection in E. coli. This is origin of replication in E. coli. And then we have this, the LTR. So in the, the whole retrovirus, we have deleted, this is a vector already. That means a lot of these genes are of the viral vector are already deleted. So we take helper cells, which would have all the genes required to make the capsid and other things. So you are introducing this genome, which will be replicated, and the capsid and other things are made from by other parts, and now this will integrate into one genome, one viral particle, and that virus particle is is used for infecting target cells. Once they are there, you'll analyze whether they are recombinant or non-recombinant and so on. Okay, I hope you have to remember about what is a helper cell. Helper cells are usually the ones that code for the rest of the genes required for formation of a phage particle. Okay, in many instances you will find helper cells or even helper phages are also there. Okay, so typically how retroviral vectors are used is you take a donor female, you take the egg cell, make an embryo, and you use the retrovirus, you infect the embryo at the 8 cell stage or so with retrovirus, and then you put the manipulated embryo into a surrogate mother, and then you would get several pups, out of which, I mean, you will isolate and find out which of it is a transgenic founder. It is still heterozygous, and one should make it as a homozygous one by inbreeding. So I think you have enough idea about what we are speaking, because we have read majority of the vectors in our things. Only thing I would want to say before I move on is that why should we not have a complete virus here? If the viral vector had all the genes responsible for making the capsid, proteins, and all those things. The target cell is infected with it. But whereas we remove all the elements required for capsid formation, replication, and other things, and we are providing those genes through the helper cells. So when the virus particles are made, they are not infective. They will integrate, and that's it. Nothing else will be done. Nothing else could be done by that virus. No further viruses can be made, right? Because it doesn't have the genes for capsid proteins and other things. Okay. Now we are having a look at the first vector, first method, where we said direct DNA microinjection. So there is typically, this is the fertilized egg. One is a female pronucleus, the other one is a male pronucleus. And then the egg is held together by a holding pipette through suction. So there is a negative or vacuum like thing, suction here, because of which the egg is held like this. There are numerous videos on YouTube if you are interested to watch. It'll be nice if you can watch, exciting actually. And they puncture the egg with lasers also can be used, because the outside of it is covered with a thick coating called as zona pellucida. Pellucida, yeah. And through the glass micro needle, they might inject the DNA into the male pronucleus. Okay. And then hopefully it will get integrated. So this is how it would look under the micromanipulator, where we will see it in the next slide probably. This is the holding pipette. Everything is done under the microscope, so that one can visualize the where injection is done and so on. This is the suction pipette, egg, fertilized egg. The outer covering, you see all these things, that is all zona pellucida. And then here is the needle through which the microinjection is performed. So this is basically same. Yeah, once microinjection is done into the male pronucleus, then it will fuse to become a nucleus, and then it's transplanted into the mouse, and then we have a transgenic mouse, the baby of it. We are already calling it as a transgenic mouse because all the cells of this organism would have derived from the zygote. Sorry. That means zygote, because as we have definition, transgenic should have all cells of a transgenic animal should have the gene transgene. Excuse me. Yeah, sorry. So this is basically the same. One important thing, however, is that we should have isolated and identified how is the transgene present or not. In the last class also, I think I told you how we should have analyzed it. One of the techniques is using Southern blotting or using PCR based approaches. Southern blotting, you should have made a probe that is specific only to the transgene. And when you perform an experiment, you should have had a control wherein you will have a normal cell, assume that is the wild type, and the other one is engineered cell. And you would have used a probe for the transgene, and wild type should not have had any band, whereas the engineered one should have had a band specific to the transgene that was introduced. Then you call it as then it is a mutant or the transgenic animal. I'll skip this because it's the same. There is a little more when we are saying it as a simple words that it is put it into mouse and so on. But when we are trying to do the manipulation in the mouse, in the animals, there is a lot more preparation to the cells. So typically the experiment would have taken something like this. They superovulate a female donor, and then they collect the fertilized eggs. Okay, they will allow these to mate. Superovulation to increase the number of eggs produced, because every time we are doing all these manipulations, we are actually damaging the eggs. In that case, not all the eggs would survive. Many of them might die out, right? So we should have increase the probability of having transgenic pups. We should increase the number of eggs that were used in the manipulation. So the superovulated donor is allowed to mate with a male, normal male, and fertilization would have occurred, and then it is sacrificed, kind of, and taken. The eggs were taken out. Eggs will be taken out, and there within the fertilized eggs oocyte, you would have introduced the transgene into the male pronucleus. Then there is also preparation for the foster mother or the surrogate mother. The recipient female, okay, this is the surrogate mother, would be surrogate mother, is allowed to mate with an vasectomized male. Basically, the female should have, there are instincts. We are all instinct-driven people, where because of several instincts, our physiology is also controlled by that. If you are reading The Origin of Species, there is a whole chapter by Darwin on instincts. I would encourage you to read that. It's very interesting to know many observations he has made during that time. So why vasectomized? We don't want any sperm to go into the female. All we need is to the female to experience the mating, so that it will physiologically get prepared. The physiology of the person in the female changes, and it will start producing hormones and other things required for holding on to any implanted embryo, right? If this process is not there, and the female mouse cannot hold on to the embryos or eggs that we are inserting into the body, its physiology should have been manipulated or modified, so that its body kinds of senses it to hold on to, or to the fertilized embryos or embryos. To do that, most natural and easier way is to allow this female to mate with a vasectomized male, right? So that mating is happening, but there is no transfer of sperm from this individual to the female. So by that event, the female gets active to hold on to the embryos, and once in that stage, we should put the embryos into the fertilized eggs into this individual, and she would then give birth to transgenic pups. And then we should have analyzed if they are transgenic or not. And if they are transgenic, we should allow inbreeding so that they will become homozygous for the transgene. I hope you understood the process of this. So now the important part is to understand what is positive selection and negative selection. We have already done this before, and I would want to repeat it once again. What is positive and what is negative selection? Positive selection is the presence of a gene is conferring a survival advantage. Gene is conferring an advantage, that is growth advantage. Okay, like for example, we have a antibiotic resistance gene, and it's in bacteria. And if we have grown this bacteria in the presence of that antibiotic, only this cell will grow, and those cells that do not have the gene for resistance would die. So here the gene is conferring a growth advantage in the conditions of antibiotic, in the presence of antibiotic, or selection basically. You can also call it as selective advantage. Gene is conferring the selective advantage. In um, negative selection, gene is a disadvantage. That means if the gene is present, the cell dies. So if we have two cells in which you have the gene, a particular gene, and the other one doesn't have, you provide a selection, and those that have this gene die, and those that do not have survive. There, the presence of gene is a disadvantage. That is called as negative selection. Presence conferring an advantage is called as positive selection. We will now we are trying to we are trying to study about engineered embryonic stem cells. In that, in the engineered embryonic stem cells, we have several steps. We will today, I will just introduce to you some concepts, and then positive negative selection. That is what you should understand. After that, we will discuss again. Okay, if required. So the first thing is I will create an objective to just to say, so that we'll have a way of learning it in simplest way. The first thing is I have a gene. This is assume this is the genome, and I will call these genes gene X and this one as gene Y. And I'm trying to establish what is our objective. So I want to introduce something, a gene between X and Y, and try to remember exactly what my words are when I'm trying to describe the objectives. Okay, this is a targeted gene transfer. In this sense, my objective in this particular engineered embryonic stem cells is that I will call this as gene W. I want transgenesis of gene W. I want that we want to introduce gene W into the genome. Second thing is the introduction should be only between X and Y. That is the second objective you can think of as. The third one is nowhere else. That means gene W or any kind of modification should not have been anywhere else. I think I should also put one more. There should not be any, I'll call this as W should not be anywhere else. Okay. And no other gene should be introduced. Okay. See, I want you to also look at this from the perspective of direct gene transfer. You should see it much more clearly, basically much more clear, because we have something here. I'll just wait a second. I'll adjust this. Yes, we have done direct gene transfer. Now I want you to compare the conditions that I already raised here. Transgenesis in direct method, what all happens and what don't happen. Direct method, um, transgene W, yes, that is also likely to happen. Only between X and Y, that is not targeted, right? We don't have a target. It's not usually not targeted, especially you're using viral vectors and so on. They may integrate anywhere in the genome. We don't know where exactly it is targeting. The third one is nowhere else. W should not be anywhere else. Because it's not targeted, it does not fulfill this one as well. And no other genes should be introduced. Any other genes should have been introduced. No, whatever the genome has, something like viral vector, if you have used, okay, they will introduce other genes also, whatever is there in the vector, and those are all the risks that are available. Something like for selection, for example, I want W, but I don't want the genes for selection in the genome, and that is a risk, right? So in comparison to direct method, engineered embryonic stem cell has several advantages, and that is what we are going to see. And that is why there is a need for engineered embryonic stem cell method. Let's try to go through this selection method, and I hope you will be able to follow it well. So first thing, we also have to introduce, we will try to learn all the genetic elements that are present so that it's easier for you. First one is neomycin resistance gene. And the second is thymidine kinase from herpes simplex virus. And we also have, this is a replacement vector. I think the objective is to put the gene, this particular element. Okay, should I call it as gene X? Yeah. So I'm going to draw again, draw here, so that it's much more clear and easier. So we have a genome, and this is the gene that I want to introduce. I will call that as X. Oh, sorry. We should have called it differently as we have told. So I will follow the same, whatever I have described here. I want to put gene W between X and Y. Okay, I'm following that scheme so that you will be able to comprehend better. So I will call this as gene X. This is the gene W. That is the transgene that I want to put into the genome. And here is, I want it targeted, right? And this is gene X, and this is gene Y. So if I want to target it, as I was explaining in the beginning of the class, we should have some regions, homologous regions to that of X and to that of Y. So I have to introduce these X and Y regions in there. And please remember, this is the first one is the DNA construct. I am going through each of the construct elements that are in the DNA construct, so that you know why is a genetic element present in there, and where is the, excuse me, sorry, and why is the where is the genetic construct present. So we are going to So if we do homologous, if we insert this gene, there are two possibilities. It may integrate or it may not integrate. How can you identify or select only those that integrated but those that did not integrate? We need something for positive selection, right? Positive selection, I will use like this here, whatever is used, I'll use neomycin resistance gene. Neomycin resistance gene. So So now those that have, if a cell has got the neomycin gene, and there is a cell that did not get the vector, okay, those in which integration happened, say integration happened here, and integration did not happen, no integration in these cells, then if I grow these two types of cells in neomycin, one of the cells will not grow, and that is those that do not have neomycin would die out. Only those that have neomycin gene along with the construct will grow here, right? That is positive selection. W by itself does not have a, if it does not have a phenotype, selectable phenotype, then it cannot be directly used. So you need to use something like neomycin phosphotransferase and so on. The gene for neomycin resistance is neomycin phosphotransferase. And that is the reason why we have introduced a neomycin resistance gene um, in the DNA construct. So I'll erase this again. Now there is also one more point is in in the establishment here, I said that there should be, we have transgenis W, we have had it in the DNA construct. Uh, Between X and Y, we want integration only to happen between X and Y. So we have put it in this way so that it is easy. I mean, W is between X and Y in the construct. Now we need to also have nowhere else. For that, we want to have an issue. We have an issue because if we do if we introduce a DNA construct like this into the cell, it may integrate here through homologous recombination. But there is also a possibility that the same thing could go and integrate somewhere else in, say, there is a gene P and gene Q. It might go and integrate there, the whole thing, X prime, W, neomycin phosphotransferase, and also the Y prime. These regions I'm just putting here. The condition that we had is we want integration between X and Y, but nowhere else, right? That is when we are going to, that is how we are going to see it. So for that, we have to do something else. That is where we introduce the thymidine kinase. Okay, this is TK from herpes simplex virus. I'll just put it as thymidine kinase. Now we have a setup that we can understand what is given in this part. So try to follow with me, and you also keep these things in mind, so that because when you are supposed to answer, you will have to do it in this way. So we made a DNA construct. It has the gene of interest. We have the regions of homology for targeted integration. And then we also have thymidine kinase here, a kinase gene. So integration can happen in two ways. Homologous recombination, it may also happen non-homologous recombination. So if it is by homologous recombination, gene W, for example, will get integrated here. Okay. And what all will integrate when it is integrating? You have to remember that integration happens between these two and these two. What is happening to thymidine kinase? It is not getting integrated. Only W is getting integrated. Look what happens if it were non-homologous recombination. The whole thing will get integrated because it's not based, this integration is not based on homologous recombination. So sometimes breaks may happen, or some other ways it can get randomly integrated into any part of the genome. And then in homologous one, you got only W integrated. In non-homologous one, you got gene W and also thymidine kinase. If I were to, so there are two total, upon integration, there are total 3 types of cells once you add the gene construct to the cells. So I will write it as W neomycin phosphotransferase gene and thymidine kinase. You assume this is one DNA construct you added to embryonic stem cells. And then there are 3 possibilities, right? Some cells did not get anything. They didn't have anything at all. Second is cells that are by homologous recombination. You can call them as recombinants. So these are, the first ones are non-recombinants, the second ones are recombinants. There are 2 types of recombinants. One is through homologous recombination, and the other one is non-homologous recombination. There is a difference in the way they have integrated between homologous and non-homologous. In homologous recombination, the gene is between X and Y. In non-homologous recombination, the gene is somewhere else, not between X and Y. And there is also one more difference. In homologous recombination, thymidine kinase gene is not integrated. Whereas in non-homologous integration, thymidine kinase is also integrated. So how to get rid of it? If you are growing, in this case, in this situation, if you gave neomycin in the culture medium, the non-recombinants would die. Recombinants would grow because either with homologous recombination or non-homologous recombination, they have neomycin phosphotransferase gene in both the cases. So they both will grow. But my desired, based on the conditions that I have laid out in the before, in the previous slide that I have described here, I don't want any integration anywhere else. That is compromised, right? We cannot be sure about whether integration is only in X and Y and nowhere else. So for that, we need to do another staining. That is, again, here is the positive and negative selection. So you have recombinants with random integration insertion, that is non-homologous recombination. These are non-recombinants which did not receive any DNA. These are recombinants with homologous recombination. And once you add neomycin, only recombinants will grow. In recombinants, we have, as I was describing before, you have random integration sites, that is through non-homologous recombination, and through homologous recombination are these. The difference between these 2 cells is that the non-homologous recombinants have thymidine kinase also integrated, whereas those that have integrated through homologous recombination between X and Y do not have thymidine kinase, no thymidine kinase. Now we perform negative selection. So negative selection here, why we chose thymidine kinase is that, thymidine kinase is an enzyme that can act upon a prodrug that is something like, I think I have already described, but I will just go with ganciclovir. It's a compound antiviral agent that has been used. I think ganciclovir, okay? And it is a prodrug. That means by itself it is not toxic as such. But if it is acted upon by thymidine kinase, then it becomes toxic. Okay? So if we have 2 different types of cells, those are non-homologous recombinants and those that are homologous recombinants. Homologous recombinants do not have thymidine kinase, whereas the non-homologous ones have thymidine kinase. And if we grow these cells in ganciclovir, okay, then those cells that have, one second, I think I made a small mistake here. I reversed it. So let me correct myself. So these are the ones with these are with the homologous recombinations, no thymidine kinase. These are with homologous, non-homologous recombinants. These have thymidine kinase. When we grow these two types of recombinants in the presence of ganciclovir, if any cell has thymidine kinase from HSV, it will convert it into a toxic compound and kill the cell. Therefore, that is negative selection. The presence of thymidine kinase within the cell is killing the cell, right? So all these non-homologous ones will be eliminated, and um, what we get are the correctly recombined, that is homologously or targetedly recombined gene construct. Okay, I hope you understood that much. I think, is there any question? I think I'll stop here. I will take up the rest of it in the next class. Any questions in this case? If you have, please ask so that I'll be able to answer if there is any doubt. Any questions? Well, I, as a part of what I was describing, we have now achieved these two using positive and negative selection. Um, this we have achieved through positive selection. That is introduction of gene W between X and Y, right? Positive as well as negative selection. I mean, nowhere else also included. We will get this. One another thing we haven't addressed is this part. No other gene should be introduced, right? What do we have here? If integration has happened, right, you will have W and also neomycin phosphotransferase gene. This is not, this one of the goals is to have W, but not no other gene should be present anywhere. It should be introduced by us. So we still have to, in a way, get rid of it. If we didn't use neomycin phosphotransferase gene, we wouldn't have been able to select at all, right? But now we want to get rid of the neomycin phosphotransferase. In the next class, we will have a look at that. But otherwise, if you have any questions, we will discuss this. I will try to repeat a little bit in the next class too. Any questions? Any questions about the paper that I have shared yesterday? Did you have a look at the paper yet? Anybody had? Hello? I have 5 minutes, right? Okay. I'll do something fun so that you'll probably do something about it. Can everyone see the, did you all get the link for the Jamboard? Yes? Can you all
Yes,
see
sir.
it?
Yes, sir.
Okay. All of you are supposed to sign on it using your mouse. Do it faster, and then we can close it. I wanted to try if it works out this way. Or you can draw some smiley also. That's also fine.
Sir, it's in view only mode, sir. You haven't gave us the access for it to edit the Jamboard.
Now it should be possible. Please check once.
Yes, sir, it's working now.
Do it faster. You only have 1 minute. Next time we will try to have some polls and other things so that we all might be able to learn in collaboration. So this is just to get started. Let's see. Are all done? I'm shutting it down.